Precision medicine is really about gaining insight by analyzing big data across populations from patients in healthcare and patients at home, taking those insights and focusing them down onto the care of the individual patient. We finally have the ability to really deliver much better, more precise care. This will make a very big difference in kids' lives. There's another piece to precision medicine, is really making sure that we have the workflows at the point of care so that clinicians can be advised and can get decision support on how to apply what we do know. I think we could move the needle very significantly in the short term. The hurdles have to do with how we link data sources together, obtain consent in a robust and protective way, how we reshape the point of care using modern technologies, but driving decision-making that is data-driven. Precision medicine is not something that needs to remain futuristic. There are steps we can take to begin to implement much greater insights at the point of care for our patients now.
The future of pediatric precision medicine. Yesterday we saw a heavy focus on cancer, and we've seen the Obama administration focus very heavily on cancer. But what makes this so unique is we're going to talk about pretty much everything else. And so each of you is very unique, a great diverse group of people. And we're going to talk about the economics of drug development. We're going to talk about costs of scaling and what each of them do individually. Because some of the most incredible advancements are coming in the form of genomics, epigenetics, rare diseases. And so what I'm going to do actually is, because everyone is so unique, let's quickly go down the line and each of you give a very short 1 to 2 sentence introduction about yourselves.
Sure.
Start with you.
So I'm Matt Might, and I'm delighted to be speaking about the future of precision medicine because I feel like I'm living in the future. My son is actually one of the first precision medicine patients. He has an ultra-rare disease. He was the very first case ever discovered of his disorder through exome sequencing, and I'd say we've been sort of unreasonably successful in improving his quality of life through precision treatments. By day, I'm a professor of computer science at the University of Utah, but I'm on leave now working with Ken and Zach as a visiting professor at the Harvard Medical School. And what's really on my mind is one question, which is how do we scale what we've done for my son for all other patients?
And I'm Wendy Chung. I'm a clinical and molecular geneticist at Columbia University, and I'm on the doctor side, a lot of the providers for patients like Matt's son. So I take care of a lot of patients who come in my door without a diagnosis, but who have some serious symptoms, and hopefully leave coming out of the door with a diagnosis, and now a lot of trying to think about how we bring this really to all the kids across the country.
Ken Mandelet, I direct the Boston Children's Hospital Computational Health Informatics Program, and we have 23 faculty determined to stop the current practice of imprecision medicine, where all patients are treated similarly, where data does not reach the point of care, and where the insights we have into the taxonomies of diagnosis and treatments often are insights that are 50 or 100 years old.
I'm George Church, professor of genetics at Harvard Medical School and health science and technology at MIT, and I work on technology development for reading and writing genomes and epigenomes, bringing down costs, improving safety, all the way to therapies, including CRISPR genome editing.
I'm Dominique Van Hollen, and I'm a strategic advisor in Third Rock Ventures. I actually came in Boston recently. Before that, I was working in Pfizer, mainly in oncology and epigenetics, in drug discovery, and before that in Celgene. So I spent my early career in epigenetics and oncology.
I'm Greg Steinberg. I'm currently head of clinical innovation at Aetna. I think it's probably fair to say that the folks to my right have forgotten more about genetics than I will ever know. But be that as it may, what we're involved in is real-world evidence generation around things such as metabolic syndrome, digital cognitive behavioral therapy, Tai Chi in the elderly. We have a lot of different things. I'm an internist and a cardiologist by training. and I'm intrigued to learn what I'm going to learn during the next hour or so.
Great. So as we talk about this gap between where we are and where we're going, I'd like to start with you, Dominique, because you left Big Pharma to come to Third Rock. And why? Can you start there with why you're excited to move and start and join this team?
So yeah, so like I said, I moved 1 month ago in Boston from San Diego. And many people have been asking me the whole month why I moved to Boston, mainly focusing on weather. The reason why I'm here is because so I spent almost 15 years to try to understand epigenetic mechanism, and mainly focusing in oncology, because behind genetic, there is another regulation mechanism that regulates why genes are transcribed. And so this is really something that we can intervene at a therapeutic level. The reason why we started by oncology is because we don't know about the safety part. And so recently, I would say the last 4 or 5 years, there is several drugs that went to phase 1 for oncology. And they actually show that even if you affect epigenetic mechanism, you are not totally messing up the whole transcription program, but you can have selectivity. So that was kind of the reason why I came here is, okay, what can we do beyond just oncology? There is like for rare disease, for example, there is cases that are pretty clear. There is some insertion, for example, of triple nucleotide sequence like Fragile X that actually, because of this insertion in the gene, is actually changing the whole epigenetic transcription, the whole epigenetic mechanism that lead to the absence of transcription. So now that we know, I'd say we don't know for sure, but we have a higher confidence that epigenetic drug can actually be safer than what we thought. I am very excited to see, can we actually intervene in this rare disease, regulating transcription through actually modifying the epigenetic mechanism by developing a drug that would affect particularly enzyme.
So you mentioned, you were talking about like rare disease and moving from oncology. I actually want to build on that and come to you. So I've actually heard you say that rare diseases are the place to start, kind of flipping this on its head. Will you tell us a bit about that and why that's your belief?
That's right. Yeah, I know there's a major focus on cancer, and I respect that. There's a huge need in cancer. But I'm telling you right now, I think if there is low-hanging fruit in precision medicine, and I believe there is, it's definitely in rare genetic disorders. With a rare genetic disorder, it's typically one gene that's wrong. It doesn't change over time. And so in many ways, it's not a moving target. It's just one thing you have to focus on. So in some ways, I would say that rare genetics are sort of the gatekeepers to broader precision medicine. If we want sort of full mastery of the genome and to be able to modify the behavior or activity of any individual gene, we're going to get there by focusing on the rare diseases that are associated with each of these genes. So because it's just one gene at a time, that's why I see rare diseases as the low-hanging fruit of precision medicine.
Nice, and you say as we get there. And so Wendy, I've heard you describe how we get there is building the plane as we're flying the plane. So can you expand upon that?
So I think the wonderful thing is we've just had an explosion and a convergence of lots of opportunities simultaneously. So we've had the human genome sequence, so we had the reference in terms of comparison. We had prices drop tremendously with next generation sequencing technology, so we could suddenly afford to do many of the experiments many of us had always wanted to do. We've got huge data sets now in terms of the clinical data to do this. And we've also got patients that are really empowered. It's sort of a new generation of patients because they have the internet to be able to connect with each other, to find each other. But as we're doing this, it's been interesting because this experiment, it really has been somewhat of an experiment of clinical implementation, is to use some of these new technologies for patients like Matt's family. But as we're doing this, we're in some uncharted territory. We're in lots of N-of-1 situations where we're describing brand new disorders, and we're a little bit making it up as we go along. It's an exciting opportunity to do it. We're learning on the fly. I think it's propelling us forward much faster. But to be totally honest with this, we are learning in the process of providing this clinical care for patients.
So as we go a little further down the panel, you had mentioned an N-of-1. But some of you are not working with an N-of-1. You're putting a lot of zeros behind that one in your case, or you've got millions of lives to cover. Can you talk about those differences?
Absolutely. So we treat each patient, again, in an unnuanced way. Let's think about big data for a minute, and I'm going to give you the use of big data for precision in an extremely simple clinical scenario, strep throat. You have a child who's got a sore throat. You want to know, do I give this child antibiotics? What we do is we take a history, we examine the throat, feel the lymph glands, and then we give antibiotics. Many folks actually use a formula, a score called the Centaur score on how to give antibiotics. But it treats each individual as if they're living in the exact same environment. So if my son has a sore throat, the first thing I want to know is strep going around in the area? That knowledge is easily gleanable. And so we did a project with Minute Clinic that actually takes uniform data on strep as one of the diagnoses they treat. Millions and millions of cases of strep. So in a big data set about something so simple as strep, we found that if you adjusted your therapies for whether there was strep in your area or not, and went under a diagnostic algorithm that took this surveillance piece of precision data into account, you save hundreds of thousands of antibiotic courses per year. All you need to do to make that happen is to bring one data element, the local incidence of strep in your area, to the point of care. Right now in healthcare, there is no systematic way to do that. So imagine the challenge we have now about bringing all this data for precision care to the point of decision making. There are approaches that we can take, and we may get into those later. But it's hundreds of thousands of courses of antibiotics saved by bringing one number, a proportion, to the point of care.
Yeah, I think from the perspective of payers, the whole idea of big data is looked at really from the point of view of, obviously we have access to millions of patients and a lot of the data behind that, which allows us to, first of all, identify large sets of individuals that fit certain large clinical profiles, and then essentially subject those individuals to some of the interventions that we're hopefully going to be talking about in some detail. We've tried to do it to some extent from the genetic perspective just recently around metabolic syndrome, which is obviously a very common problem. And one of the biggest issues around metabolic syndrome is, not surprisingly, the whole issue of weight management. And there's no dearth of potential solutions out there to dealing with weight management. The problem is that they suffer from poor engagement. So most people just don't persist. And one of the theses that we were trying to look at was, could you use a sort of genetically guided approach to help personalize an interaction between a coach and a potential patient to allow for greater engagement and ultimately greater weight loss? So, I mean, this is an example of how, again, large data sets can be merged with the concepts of precision medicine to advance overall health.
That's great. So I'm going to switch gears a little bit here, because as you let in, you said payments. And I immediately thought, so great, how are we going to pay for this? Let's talk about that. And George, you've been quiet, so I definitely want to get this in the mix about how do we scale? How do we lower the cost and scale these things to populations?
Well, you can be hopeful based on history, although it's not necessarily predictive, that we've brought down the cost of the genome sequencing from whole genome sequencing from about $3 billion to $1,000 today. That will continue to plummet. Finally, there is some non-monopoly rumblings in China that will might aid this if Americans follow suit. And it's extraordinarily important that we bring that down to a point where everybody has their whole genome sequence, because it's predictive not only of regulatory elements outside of coding regions, but the coding regions themselves can be disrupted by structural variants of various sorts. So So that's one part of it. But then there's a question of bringing down the cost of therapies. And one way to do that is via preventative strategies. Mm-hmm. I think, you know, a huge fraction of cancer that we heard about yesterday is focused on curing it, but there's also, most of the real breakthroughs have been in preventing it, whether it's removing organs at risk due to genetic risk factors, 11 different infectious diseases, and so on. So I mean, I think that's one way we're going to really bring down the cost is via prevention. It's hard to get a new prevention through the FDA if it is at all invasive. Mm-hmm. If it involves walking a little bit more or eating something slightly different, that's okay. But yeah, anyway.
From a mechanistic also point of view, to decrease the cost, now I'm talking about the cost to develop a drug, which is actually kind of linked to the cost of a drug. Right now we talk about $1 billion to develop a drug. I mean, the more we know, the more we understand the mechanism, the faster and the more precise we can develop actually a drug that can intervene in the mechanism. And hopefully, instead of spending from an idea to a drug, even in clinic and then marketed, instead of spending 10 years, can we spend 5 years? And then the cost of developing the drug will be lower, and pharma or even biotech would feel less empowered to ask a high cost for this drug.
Would it be half the price, or would it be even lower? I mean, if it's half the time, is it half the price? Or is it...
So imprecision medicine is also costly. I published a paper earlier this year looking at screening mammography. And if you look just at the false positives from screening mammography, and you look at the overdiagnosis, some women end up being treated for cancer who do not have cancer. That costs $4 billion a year. That $4 billion a year put into a much more targeted screening program would be one step towards precision medicine, and the cost savings would be tremendous.
Wendy, can you give us a specific example working in autism of issues of this right here?
Right. So autism, as many of you know, is pretty common. 1 in 68 children these days are diagnosed with autism. But in fact, autism is not really autism, it's really the autisms. And current estimates are probably that there are at least 500 different genetic causes, in addition to other sort of exposures in either in prenatal life or shortly after birth. When we think about this, really there's probably definitely more than one neural mechanism in terms of causing this, and it's very likely there's going to be more than one treatment. And beyond that, it's not as if all of this is going to be pharmacological treatment. A lot of this is going to be behavioral treatment and other supports that we're going to be giving families. But one of the things that I think, as we try and scale these, we had talked about prevention. Autism is one of these things. It's a very classical thing in my mind as a pediatrician with prevention. Children are developing. That's what's neat and different about children than in our adult populations. It's very dynamic, and brain development is especially very dynamic. And so if you make an early diagnosis, you can amplify the effect of what you can do if you can set the brain on the right course from early on, rather than waiting and trying to fix things after the highways are already built, after you've already got myelination of the brain and all of your networks. And so one of the things we're trying to do within autism is actually figure out how to do this and to scale this. And as we're doing this, we're trying to figure out how we can do this with really engaging the community, doing this as a partnership. So we're actually going to be starting in April 2016, you heard it here first, a nationwide study to actually engage 50,000 families across the country with autism, with one center right here at Boston Children's Hospital. And we're going to be doing this to study their genomes. In addition to, it's not just their genetic information, but what happens on a daily basis with the challenges with behavior, with learning. What changes with your sleep and how your child behaves, what happens with what they eat and how your child behaves, but trying to ultimately use that information, not because genetics is the only thing, but it's an important anchor we can use to get into the biology, to understand everything from synapses, neurotransmission, how the brain develops, to understanding those things to come up with better targets. And hopefully, as we engage parents, to really think about patient-centered or parent-centered outcomes in doing this. Behaviors are things that matter most to families in terms of what they'd like to be able to modify. So I hope we're going in directions of things that matter from the individual's point of view, rather than something that the FDA has approved just as an outcome measure because we've tried it before.
Did you have something?
Yeah. So I'd like to add sort of the rare disease perspective on the issue of cost. So there's some 7,000 rare genetic disorders, and there's more being discovered all the time thanks to next generation sequencing. And if the cost of developing drugs really stays around $1 billion, we're never going to get to a cure for all of these diseases. So I think in the short term, what we have to focus on is leveraging the arsenal of drugs we already have approved with repurposing. And I think this is a space that we really haven't tapped into in a meaningful way. And I can give you some examples of where this has been successful. So there's a lot of novel genetic epilepsies popping up around ion channel disorders. And there's already a lot of drugs that manipulate ion channels. They're usually cardiac drugs. So now you're finding these N of 1 cases that are treatable for epilepsy with a drug that was meant to treat the heart. And I think we're going to see a lot more of this going forward. But it certainly opens up some questions like, well, how do we know? How do we map the right drug to the right patient over and over again when it's so individualized? And how do we know what we're doing is safe? I think that's a big question.
And in addition to that, I mean, one part that is also missing, in oncology, it's easy. You treat a patient, you look at it, the tumor shrink. You have a very clear biomarker. For some of these diseases, it's not clear what to follow, because it's not like suddenly you're going to have a child that's going to totally revert his behavior because you give a drug. But you want to see if there is any effect of your drug. Mainly in clinical trial, it's like, I mean, are we improving something? Are we expecting a cure? Probably not, but something that can help them to confront society or to be more independent if it's CNS link. So the study that you are mentioning, and I'm sure there is much more study out there, are going to actually provide information of all these different biomarkers that we call that. It can be development, it can be molecular, or it can be just behavior that can help us to say, are we improving something with a drug that we are pushing forward? So this is also a very important part to try to go into this precision medicine and understand better.
So you said behavior, and you talked about engagement, and we hear this word engagement everywhere right now. But to get all these people enrolled, you actually have to engage people, which I'm sure is something you deal with all the time. How do you make that happen, especially in such young and vulnerable populations?
Well, I think that engaging, having parents and children be engaged in some of these issues that we're talking about, I'm not sure that's the challenge, because they're very motivated for the obvious reasons to do whatever they can for themselves and their children. I think the bigger problem is one that it goes back to the cost, because at the end of the day, we actually are not dealing with an infinite source of funds. It is finite, and it's coming from all of you guys, right? Again, you know, the tax base, right? It's not being generated somewheres in somebody's basement, right? There's no printing press out there. So unfortunately, sooner rather than later, we as a society are going to have to come to terms with making some decisions about what do we want to go after and in what order. Because you can't go after everything at the same time. It's not possible. It's not possible from a resource perspective. It's not possible from a financial perspective. And we have to wrestle with that and come to terms with it. And it can be done. I mean, other societies are figuring this out. So the Brits are figuring this out. They have a national level commission that does just that. They look at therapies and they compare them in terms of cost effectiveness.
Okay. Do you want to build on that?
I just wanted to say engagement, I think everyone's heart is in the right place. I think life gets in the way, though, and especially when we think about children, in particular in children's health, and children especially who have chronic medical care issues. Life is complicated and it's quite challenging sometimes. And so I think part of our solution has to be in this to make it easy. So if someone wants to engage, whether it be on the provider side, whether it be on the patient side, make it as easy for them as possible, and really make them part of the team. I mean, I think it's, you know, one of these things we've done in research, and I will put my research hat, is we've made this a one-way street. We've made this, you know, information coming to the researchers. We've made this about people building their careers, and that's not what it's about. I mean, it's about trying to move the ball forward for patients and families, trying to get them some short-term gains as well as the very long-term gain. And I hope we're entering a new era where we're really trying to do much more of that. We're trying to feed this back in real time, and we're really trying to make a difference, not in 10, 20 years, the next generation, no, for this child today, tomorrow, but make it really about the care as well as the care until the cure.
And it's also important to explicitly acknowledge the somewhat artificial divide between research and clinical care. In a health system that is healthy, every time we have a new outcome related to a therapy or a trajectory of a child with a rare disease or an adverse event related to a drug, there's a kind of an assumption among the public that the health system is learning from that and that the very next patient treated will not be treated in the same way as the one in whom something went wrong, and on the other side, the ones that do well will get more, future patients will get more of that. And so we really need to instrument the delivery system to be able to follow people. That's how you get cost efficiencies. We're already treating people, where we're already documenting in electronic medical records, submitting quite detailed information about diagnoses and ultimately medications into are fully known by organizations like Aetna. And the opportunity to take the delivery system, to use it not only to collect data, but also to do experiments within the delivery system is another way that we can get this done. Think about it. If 50% of kids are on one ADHD drug versus 50% on another, and it's just random based on, for example, the provider's preference. There is the opportunity to actually, instead of allowing this random 50-50 to happen in a way we don't control, to actually randomize 50% of kids to one drug and 50% to the other within the healthcare system, and then without any additional research infrastructure or research dollars, we answer the question as to which one of these drugs produces a better outcome.
Good.
Yeah, I think we've been talking about increasing participation and the complexities of families that are already affected. But what about increasing participation among unaffected individuals in the world? And we're talking about reducing costs by bringing the price per drug down from $1 billion to $500 million, or reusing drugs.
You didn't say that, bud.
I know, I said that, but or to 400 million even. But the alternative is many of these things have existing technology that works on people that are unaffected, and that is preconception and prenatal diagnosis. Very cost-effective, very inexpensive, almost never practiced unless you have a pre-existing genetics in your family. While a priori, it's very likely that you will be the first. When two recessive individuals get together, they could be the first in their family, or if there's a de novo mutation, you will be the first in your family. So why is it not the case that every person at least in industrialized nations, where people think very little of adding an extra $1,000 to their car price or their house price, and they don't get reimbursed for their car or their house, right? Why is it that they're waiting to get their own genome sequence?
So I'll just amplify this. So I think that's eventually the goal, and I hope people can get their heads around this. I think we're, as the analogy of building the plane as we're flying it, part of the problem for us right now is just interpretation of all those genomes. We can't do that for the techno-speak of you out there. A lot of missense variants, for instance, I can't, and maybe George, you're better at this than I am, but I can't perfectly predict all of these things, and I'm gonna put a plug out there.
Can you perfectly predict blood pressure?
No, I can't, but.
Why are we treating that different?
So all I'm saying is we get some information, but I can't squeeze everything out of it that I wish we could now. It's not to say that we shouldn't start, but just to say that we're not quite perfect at doing this. And I think the public needs to acknowledge and realize that we are still in a learning phase of this, that we're not perfect at what we're doing. It should be the goal. But the plug I wanted to make is that and I come from New York City in terms of where I practice, part of the problem that I have with my patients is they come from around the world, all four corners of the world, and I'm getting really good at understanding the genomic information. If you're of European ancestry, I suck right now, to be honest, if you happen to be one of my Dominican families or some of my African-American families, and that's just not fair. I mean, in terms of the gap that we increasingly have in terms of genomic medicine between the haves and the have-nots, and making that gap even wider because we're building this wedge, that's something I think we as a community need to address to make sure that we're doing right by all of our patients.
Mm-hmm.
And I can follow up on that because I've actually been in the situation of having a variant that you don't know what it does. You know, when they told us that, well, we think these mutations are causal, I said, well, that's what we think. It's not what we know. And the way I resolved that uncertainty was by turning to the internet. You know, I wrote a blog post that described the whole odyssey we'd been through that was designed to find any other patients out there that might have this same variant or these mutations in this gene. And it worked. We found other patients. Then we knew for sure this was definitively the cause of my son's disorder. So there's sometimes, you know, things like the internet, it's an enabling technology that can let us sort of short circuit things like variant interpretation. And I think over time we'll find ways to scale that process.
But then the next step is to act on it, right? From a drug discovery, and it's like, okay, now we know, what can we do to improve this patient, right? It's like, it's nice. I mean, it's a good start because this is how we understand what's going on, and then we can develop a drug, and then we can treat this patient. But it's like, yeah, now that there is mutation, what can we do? And I mean, acting upside of it or downside, regulating transcription, which is what we are hoping to do with the company I'm building right now in Third Rock, which is, can we use this epigenetic mechanism to reactivate or to bypass the mutation and still have a protein that is there, functional? And I think there is hope there. And in one way, if we are there, it's because of all this genetic activity, which kind of give information about where to look at least, and not just trying to look at the kid and say we can't do anything.
My question is, why do we have to wait until we understand everything, as Wendy says, not to start, but actually to finish sequencing everybody on they can afford, that's in industrialized nations? And why do we have to wait to develop the drug if we have the technology, which is genetic counseling? That's the question, is why wait to finish, not to start?
So I mean, in my paradigm, this is an iterative process, right? So we got to start because we've got to get a lot of the kinks worked out in the system. I'd just like to make sure that the public understands that we're continuing to get better with this and that we will continue to give out information over time as we learn more. We're working together on this, and this is a cyclical, iterative process. It's not a one and done. That's the only thing I want to make clear in terms of the public, is the realization we don't have it all worked out yet, but by working together with this and continuing to engage. This is not like this type of thing, you get your CBC once and you're done with that. This is, your genome hasn't necessarily changed, but our interpretation of what it means for you to enable you is gonna change over time, I think. I hope we're gonna get smarter.
So one of the things we kind of continually heard here, and you talked about a gap in communication at this point. You talked about a gap between the haves and the have-nots. At the beginning, I talked about the gap between where we are and where we're going. So as you're having this discussion, I had to put on my policy hat for a moment. And we're really, really good at regulating ourselves out of like every good idea we've ever had. I get told all the time how good we are at screwing things up. So whether it's state level or federal, what are these hindrances that are keeping some of these things from happening? Because I assume part of it is our fault. What are we doing wrong? What would make your life better, easier, move things faster, make it cheaper?
The risk. I mean, I would say I'm coming back to my drug discovery, but the risk, your level of tolerance for risk. I mean, if we want to say, well, we don't have to know everything to start something, we need to go together. You have to be able to move forward with a belief that is not 100% sure. Mainly if we want to go in this very early new way to treat patients. And I think the human nature is people don't like to fail. And so if you go with something that is highly risky, you have a high risk to fail as well as succeed. But so I would say that is one maybe in human nature, to maybe wait and have a whole package and then move forward. Mm-hmm. Or you meet people that are okay to take risks because they want to make a difference.
Another area where there is regulation that can get in the way is the oversight of clinical studies by IRBs, and there are some barriers in there to patient-driven approaches where the patients are willing to give up some privacy or to share data and actually end up being told that it's not ethically permissible under the regulatory structure. So I think the way that we regulate research now is very dissonant with patient-driven initiatives like MATS to be able to obtain the data they need, to share the data they need, and to engage in the N-of-1 type experiments that they need.
Well, case in point is I've still been denied access to my own exome.
Case and point.
And virtually every patient we've discovered so far can't get access to their exome, which means we can't do studies to look for modifiers of the phenotype because we can't coalesce all that data together.
Yes.
I should just point out that this is not a uniform problem with IRBs all over the world because there are IRBs in 4 different countries now that approve the Personal Genome Project, or the equivalent of IRBs, which is the most dramatic sharing protocol that you can have. So it's a non-uniformity of IRB protocols, I think is the issue.
I'll just put out one of the biggest things I think that from my end as a diagnostic side is actually the FDA potentially. So as some of you know, the FDA is coming in, starting out because of their experience with 23andMe and trying to decide if they're going to regulate genetic and genomic tests. And with this, the greatest concern I have is, as I said, this is to me still dynamic. And what happens with the FDA regulation of any of these things is they literally lock down whatever it is you're doing, whether it's data generation, data analysis, but they lock it down and you're frozen and you're stuck in whatever that is. And we are just moving so fast in this field right now that we would do a disservice to ourselves if we did that. And that's a real true threat in terms of being able to implement this in the very near term.
Yeah, I also have some thoughts about the FDA, but more on the clinical side than the diagnostic side, because I feel that repurposing medications is such a huge part of precision medicine, particularly for these ultra rares, where even if you were to develop a drug, you'd never have a population size to get the statistical significance you need to prove that it actually worked. So I think what we need to do in the long run, at least for these ultra rares, is decouple safety and efficacy from approval, so that once it's through safety, where we have a sense of what the side effects are, it should be available to the ultra rare patients who are almost certainly never going to have the resources to push it through a clinical trial to show efficacy. I think that's just the reality of the situation.
Did you have something?
Well, yeah, I mean, again, I hate to keep coming back to the cost issue, but I think that's probably why I'm on this panel, so I will play my role. Part of the problem also, and look, I think we're at a tremendous inflection point in sort of medical history with all of these developments. That said, again, there is a real potential threat if this is not handled correctly that you fiscally blow up the healthcare system. I mean, just to do, even at $1,000, right? You have 350 million people as far as or thereabouts in this country. So I think that's 350 billion straight off just to have everybody genome sequenced. A significant subset of those people are going to want and demand subsequent follow-on testing after those results are in, some of which will be warranted, some of which will not. There's going to be downstream treatment, and all of that is going to snowball, and unless and until that process is understood and in some way shepherded intelligently, you have a real potential problem that you've created. So I'm all for it, so don't get me wrong. I really believe that's the right way to go. It just needs to be done in a way that is thoughtful and considerate of the downstream implications, both clinically and fiscally.
So let me say that you could start, though, again, if you're thinking about kids, you could start with kids who are symptomatic. And again, if you think about the fact that kids early intervention in whatever the disease process is likely to have greater downstream payoffs if you diagnose early. Start with the kids in terms of doing this, the kids that have symptoms, and get to the diagnosis as quickly as you can. Don't do biopsies, invasive biopsies, imaging, hospital procedures, things like that. Just get to the diagnosis. And I think some of us know that certain kids are gonna have a genetic disorder. It's just a matter of finding out what it is, and at least skim that cream off the top. At the very least, if we could start the discussion there, I think we could move the ball forward very significantly.
Yeah, I mean, there's a huge cost to being undiagnosed. For 4 years, when we were undiagnosed, we were sending the insurer $100,000 a year in bills for what you would call just sort of a fishing expedition, just lots of deep phenotyping, imaging, and then we get sequenced, and overnight we have an answer, and all that goes away. You know, so our costs drop dramatically after sequencing. So there's definitely a population where today you could save a lot of money by sequencing early.
No, and again, let's be clear. I'm not against that kind of issue. What I'm saying is that the flip side of that coin, sadly, is that you create a lot of genetic information on a lot of people who now don't know quite what to do with that information and are going to wind up doing stuff, some of which might be actually physically harmful to them, on top of everything else, and expensive. So it just needs to be balanced, is what I'm saying. For certain individuals, this is absolutely, it's a no-brainer, probably, you know, the way I would look at it. I think it's when you take a population view that things start to become a little more murky.
I think that that the $350 billion price tag is a little bit misleading, in the sense that the difficulty of drawing a line where above is something that we can definitely do something about, and below is murky. For example, with Tay-Sachs disease, to take a very specific example, that's not something where you wait until the child is born. That's, you know, Matt, you could have had a diagnosis much earlier if the entire population were screened for very highly deleterious mutations, like Tay-Sachs. It's been reduced almost to non-existence in the populations that adopt this and you can just say, if we're going to be providing the money, then we're going to draw the line here, and we're not going to be looking at you know, silent mutations in the Tay-Sachs disease. We're going to be looking at things that affect the protein. And it's not negotiable. If they want to go off and pay for it themselves, as is done in many countries, and even in the United States, then that's their business. But if we're going to be paying for it, we can get that money back, because the cost of dealing with a Tay-Sachs individual, both to the family psychology and in terms of real money, is significant. The cost of orphan diseases is $1 million or more, whether it's gene therapy or protein therapy, what have you. You can get your $350 billion back right away, as long as you're tough about drawing that line about what the interpretation is.
But I think we're agreeing vehemently. It's
yes,
just,
we are.
Right? But you just went through that calculus, right? It's how do you?
But it's been done for a variety of diseases, and we don't have to deal with the diseases that we are uncertain about. We can deal with the ones which we are certain about. Mm-hmm. And that is, I would argue, is cost effective. I'd love to hear an argument why everybody in the United States isn't sequenced today.
It's interesting, you know, the payment models are going to have to vary too, because if you look at healthcare itself, healthcare is used to being reimbursed handsomely for every diagnostic test and every procedure. And the exome, even if we had the payment for the initial exome, that's a test you do once. And then you have all this interpretation later, for which there really isn't a good model for reimbursement as well. So it actually, the payment models for this are quite complex.
But you don't have to pay that, right? I mean, you don't have to pay for something in the far distant future. You know, if somebody discovers in the far distant future there's a new interpretation for blood pressure or CBC, you didn't have to build that into the original model that provided that. If there's a $300 or maybe even $100 exome, why are we fretting about imaginary interpretation problems in the future. We've got some real interpretations that we can do today on Tay-Sachs and similar diseases. We're letting the unknowns, the things you can't do for blood pressure and CBC, stop us from doing what we can do, which is usually what medicine is about, what you can do, not what you can't.
Excellent. Well, timing-wise, that's also perfect, because I was going to say soon, I think we should start taking some questions from I'm sure they have a lot of questions. I have plenty more myself, but I want to take some time. We have microphones going around, so we'd like to start with, there are hands up everywhere. Okay, let's start right there. Sure.
So I just wanted to add a little bit more fuel to the fire.
Yes, please.
First, the cost of developing any drug, whether it's an orphan disease or one for millions of people, it's over $2 billion. That's just the average cost of any drug. No pharmaceutical company or biotech company prices their drugs based on the cost. It's the value delivered, it's the outcome delivered. And it's a judgment call, because when you typically launch a drug, you haven't even got a clue how many people are going to get treated. If you listen this afternoon, it's going to be a great session on cystic fibrosis. We knew about that gene 25 years ago. It was sequenced. Today, it took 25 years to get the first drug for treating about 4% of those people based on the particular genetic mutation they have. We now know there's over 200 variations of mutations in that gene that cause cystic fibrosis. Every one of those needs a separate drug. So it's a big, big problem that we have, and I completely agree that as a nation, we have to think carefully about what we do and how we spend our money, because there's some great opportunities, but it's very, very complex.
Thank you.
But instead of 200 drugs for cystic fibrosis, how about genetic counseling? The answer was, her statement was absolutely.
Or what about CRISPR?
My question is about the interpretation of whole genome sequencing, or any large-scale genome sequencing. Two things that I haven't heard was, one is data sharing. It's still siloed, and the more we share, the more we'll understand about how to interpret. But also phenotype and genotype correlation. Clinical labs have very limited phenotypic data, and that we haven't figured out yet. And so I'd love to understand from the panelists what your thoughts are on these 2 points.
Ken, do you want to start with the sharing?
The sharing is quite interesting. There is a whole spectrum going from the Henrietta Lacks story, where there was a sense that cells were pressed into the service of science and profit without the consent or knowledge of the individual. And then there's, of course, this imperative to have very large numbers of exomes so that, and whole genomes, so that we actually can understand an individual variant. You can't understand an individual variant in many cases and its contribution to disease unless you understand the full variability. Otherwise, you will pick up false positives, things that appear to be causative within population because the population had the disease. And so, if you find mutations in a small population that has a disease, you really don't know how many people who don't have the disease have those as well. So you need the big data sources, and the sharing is key. There's a new federal effort to actually change the common rule that enables broad consent for really all future uses of biospecimens once they're collected. This is going to be very important, but also will need to be balanced ethically with what the individual is truly consenting to and how well they understand it.
And I'll just amplify, because we're talking about kids, is that one of the things that is important is to get kids' permission, especially as kids become an age of which they can understand this and be able to give that permission to actually engage them in this too. I mean, I think the genotype-phenotype correlation is incredibly important. It's just so multidimensional that it's just a matter of big data. I mean, to be able to get really all of the genotypes you'd want in the background of the genome, it's a matter of just numbers. It's a numbers game.
I would even add, actually, on the regulatory side, I was shocked the first time I had to actually start dealing with FCC and SEC, thinking about the sharing of data and what we have access to and who should have and who can merge it, and then once it's merged, what apps it can be sent to, and so it's not just like an FDA issue. This is federal, government-wide, in lots of agencies right now.
Yeah, I think we also have to acknowledge that as researchers, there are academic incentives that lead to siloization. You know, we hoard our data so that we can be the first to publish. And I've worked with multiple rare disease patients now where to break through the silo, we had to turn to the internet and social media to find a matching case because this data was being siloed by researchers trying to publish it.
Well, at least it was a short time because it was until they publish it. Sometimes it's in industry, it's even longer because this is the value of the company to develop a drug. So it can take even longer.
And I think Matt's point is also critical. Some of this stuff is going to start to happen quite outside the healthcare system as well. George, you work primarily outside the healthcare system. And this 50,000 patient study is going to be outside the healthcare system. So I think there are imperatives on the healthcare system to stay in the game. And by staying in the game, they're going to have to compete in research with what's happening outside the healthcare system. And again, it's really the regulation, the data grabbiness at every level. Folks think that they own the exome. So the patient believes they own the exome. I think there's a pretty good argument there. Whoever the institution, if it's a hospital, believe that they own it. The individual clinician whose patient it is may believe that they own it because they ordered the test. So you have this kind of fractal level of strange impressions of ownership that go all the way down to the patient. And this definitely is a barrier to sharing. This is why you see patient communities forming completely external to the health system, and they're getting real things done now. So there's an imperative, I believe, on our institution and others to embrace patient communities, to embrace patient-driven medicine, and to figure out what its relationship is to the healthcare delivery system.
Go right over here.
I was just curious on a comment that George made that everybody should know their genome. Would we do it like a metabolic screening? And to my knowledge, something we do to every newborn is we'll get a metabolic screening. I don't know that it's ethically that things such as Alzheimer's, which I'm sure has a genetic component, or other things like BRCA gene that people might not want to act upon, why should they, what good would a genome do? And is it for everybody to cross the board? I don't think that's a realistic expectation. Second question, just a little different, was are you saying that now genetically things like cardiac disease or renal disease, I've had experience with nephrotic syndrome, that if we know the genetics, do we have to do a cardiac cath anymore? Do we have to do biopsies anymore, because we'll do, instead of doing that, do genetics of those disease and avoid those things? Two separate questions.
Yeah, in terms of the first question, I'm not suggesting that everybody should be, I was very specific. You draw a line where you're talking about actionable and predictable things. So every newborn gets tested for PKU and 40 other things. You do not include things that are not actionable. But there are things that are actionable before you get to a newborn, say preconception, which are not included in the newborn test, and could be the same kind of strict line you have for newborn testing, you could have for preconception and prenatal testing. You don't include Alzheimer's until it is actionable. You include things like Tay-Sachs. So newborns, PKU, preconception, Tay-Sachs. It's not that complicated that we have to put everything on hold or call it a start. It's already started. Tay-Sachs is almost gone. Let's expand that to a few other diseases by doing the whole exome for the whole population.
George, I agree with you that we need to move forward and keep momentum, but is it realistic to think that once what what we're all arguing here is essentially a diagnostic test of sorts. Is the whole exome is both a diagnostic test and a platform for other diagnostics to be developed against. But once that exome is out there, how do you limit its interpretation to just the actionable?
How do you limit newborn testing? You don't give it to them. If you pay for it, you could do whatever you want with it, right? If they pay for it, they can do whatever they want with it. And I think it's their right to do it both ways.
But what would be the obligation, though? So it's out there.
Obligation?
It's out there. Someone decides we're not going to test for these three things. Then a patient has an outcome. And then someone, probably a lawyer, will come along and say, but that was sitting right there.
Well, a lawyer could take the blood test from the newborn screening and say, if you tested that blood for one more thing, you would have found this, okay? But they don't do that in practice, okay? You can if you want, you can shred the exome as soon as you've gotten what is currently best practices, right? It's gone. That was the deal. We're going to sequence your genome for free. We're going to analyze what's available in 2016, and that's it. And then we're going to destroy the rest, if that's what it takes. But the point is we can do it today, right? For Tay-Sachs and 1,000 others.
We clearly have questions for each other, too. So, but I'm going to go back to the audience for a moment.
I'm going to actually shift the discussion to something a little bit more practical about some of the pressures we're experiencing in the front lines of taking care of pediatric patients. And what we're seeing is there are already patient-centered awareness about the availability of genetic testing without an adequate clinical genomics infrastructure to support clinical care teams to identify the best value genetic tests available to make these diagnoses in a more timely fashion and be more precise with management. Also, another thing is that contemporary trained genomics counselors are becoming a little bit more challenging to recruit because they're finding positions in laboratory-based in these genetic companies, and also the support by leadership to fund genomics-trained genetics counselors at the places that we're taking care of these kids, and developing an infrastructure is needed. Just wanted to bring that up.
Thank you. We only have a couple minutes left, so quick questions.
Just to build on that very succinct comment, I'm a genetic counselor from Emory University in Atlanta, Georgia. Thanks for the plug, Dr. Church. One of the things that we do need to figure out is how to pay for the genomic process, the genetic counselors and the medical geneticists, because right now there are not enough medical geneticists because they don't get paid well. It has to be a labor of love. Genetic counselors growing in numbers, but still very small in numbers. And it's hard to get a genetic counselor paid for. What we do is interpretation, which is much cheaper than an exome or a genome. But there has to be there has to be changes to how we do payment and insurance and structure so that we can interpret this data and compensated for the time that's spent doing it.
Okay, any last minute questions? Yep, over here.
Yeah, I'm a pediatric orthopedic surgeon at Children's, and I would be confronted routinely now with a set of complex deformities. The child walks in, family comes in, and they say, we have X genetic abnormality. There's only 3 people that have this disease. Now, what are you going to do about it? Now, so the information that comes is from these other families, and you've touched on this. So it's not available to me in any way. And so here's this whole HIPAA violation, medical records. I can't take a medical record home without violating rules, just a little piece of paper. And yet, the information that I need is somewhere out there on the internet that everybody else can look at in some way. So what's going on with the whole legal structure, HIPAA violations, and my access to the information I need to take care of patients?
As I understand it, the answer is in the Personal Genome Project, we take HIPAA EMRs and turn them into personally controlled health records. That is, if the patient then wants to share those, the patient can share those, and the recipient is not in violation of HIPAA.
First, thank you so much to the panel.